Good evening, everyone. Good evening, everyone. At the outset, I'll welcome our guest speaker, Professor Bala Ratna Sabhabadi from Florida. I also welcome all the participants. I welcome all the organizing team members. I am very proud to say that our guest speaker is one of our alumnus from Annamalai University. He did his UG from Annamalai University and completed PG from IARI and PhD from Canada. He has received many awards and fellowships. So far, he has guided 3 PhD scholars and 7 PG scholars. His area of interest is mainly capsicum breeding, particularly the nematode resistance, and he has also worked in heat tolerance in rice and maize. He has published more than $9 lakh as external funding and more than $2 lakh as internal funding. Totally, he has published more than 11 lakh US dollars as for research projects. He has published so far 99 research papers with a Google index of more than 6,000 and H index of 43. On behalf of the organizing team, I wholeheartedly welcome our guest speaker. Good evening, sir.
Thank you, Dr. Prakash.
Thank
you. I'm really happy to be here, and I wish we could be there in person, but at least I'm glad we could join by Zoom. First of all, I want to thank you, Dr. Prakash, and the Faculty of Agriculture to invite me to make a presentation at this event. Before I start, I just want to say 1 minute about my experience at Annamalai University. In the 80s, when I did my bachelor's degree in agriculture, I just want to say Dr. Samandam was the dean, and he was in the botany horticulture section. And one of the first horticulture classes I took was with the Dr. It's a PK, we used to call him, is P. Kalyan Sundaram. And he had an excellent teaching of horticulture, so I'm really blessed with that. I also had the opportunity to study under Dr. Ganesan and Dr. Easwaran in the botany section. And also at the time, Dr. Narayana Prasad in microbiology and Dr. Chandrasekharan in soil science were all influential, and they really taught us really well. And it was really a pleasant experience, and I used to do a lot of insect collection for entomology and plant pathology. And I remember Dr. Nachiap and Dr. Narayan Swami. Everybody, soil science department. It was really wonderful experience for me when I was there. I continue to have some association with Annamalai University and used to visit every few years once. Even though we are in the US every 2-3 years once, I come to Chidambaram because I'm originally from Chidambaram. And I was a day scholar when I studied there. So that formative experience was really key for me to get interested in agriculture and in plants per se. And then later I started doing all kinds of research. And Dr. Prakash mentioned I work on capsicum breeding, but I didn't start out as a plant breeder when I started my program here. So my area of expertise is metabolic biochemistry. So I have done a lot of work on metabolic biochemistry. However, at the University of Florida, I have started teaching horticulture. And then some research I started ended up becoming a research project that is a plant breeding research project. So my plant breeding research was actually a part of a teaching effort, and that's what I'm going to share with you today. I'm going to share a screen, and then I will use a PowerPoint presentation to go through some slides. Can you all hear me? Is there any problem in communication, or you're all okay with it? if somebody can tell me whether you can hear my audio clearly.
It's clear, sir.
Okay, very good. Thank you. I just need to, I need to do screen sharing, and if you could make me a co-host, that will be great. I think while they are working, I can, yeah.
Just a minute,
Okay.
sir. Just a minute.
No problem.
Yeah, now you can share, sir.
Sure, thank you. My title is Genetic Improvement of Peppers for Fruit Quality Traits, and this is the title of the presentation. But a lot of different people are involved in this research, so I kind of listed some of the key people, some of the students who worked in this and collected some of the data. So I put their names here. And as I said before, my plant breeding program in peppers have started out as an effort to introduce research to undergraduate students. Most of the time, the students were studying for a bachelor's degree or undergraduate students here, and they come in and take classes, but they could also do some research on the side. And they do come into different labs and they can participate. So I wanted to have an experience for students to do their own research project. And that's where I started out. And we started out this idea in a classroom because I was teaching some plant breeding in a classroom. So we kind of gave a name, really audacious name, Building Better Peppers, as though the peppers in the market are not good. We got to make it better. Because one of the goal is to breed varieties that are better than what is already there. And this is like a product development goal for the project. So we all like a lot of food that is kind of sugary and sweet and all that. We wanted to make a goal that can we breed tasty, nutritious, and colorful, attractive peppers that's going to outcompete other food. So that is the idea, that we wanted to have a goal to make fruit that is quality-wise very high quality, so that people will like. My initial work was that thinking was that maybe we will make sweet peppers, because that way people can eat it like a snack, and it'll be a small size pepper. So it's easy to package and put in the refrigerator, those kind of things. So that was my initial idea. However, much later, we kind of came up with this ideas that a lot of biodiversity in capsicum or is outside the sweet peppers. So we wanted to look at all other different peppers. Why did I choose peppers? I like them. That aside, this is a very important crop all over the world, and we have the fresh peppers and dried peppers, as you all know very well. In the US, the market is very high for bell peppers, so that's where most markets are. Chili peppers are consumed much less. And in Florida, the bell pepper is an important crop, so we are growing in 17,500 acres. It's not high acreage. However, it's production-wise, it's a very highly tuned vegetable production system where they have not only peppers, but other vegetables in cycles. So it's an important commodity. But as as a scientist, the challenge working with the peppers is that it's got a very large genome size. So I put in here 3.3 GB, and compared to tomato, it's only 0.76 GB. So it's almost like 3 times more size. And we don't know why, but we have some idea why the genome size for pepper is very large. It also has a lot of repetitive sequence, 81% repetitive sequences in the case of pepper. In the case of tomato, it's only 61%. So something happened in the evolution of the pepper where there is a lot more transposon activity that also resulted in a lot of duplication and repetitive sequence that are generated in the genome. So this is, if you're a genomic researcher, this is a challenge because you have this unusual situation. But also if you want to transfer genes into pepper, it is difficult to make transgenic plants. We do that in our lab, but very low efficiency compared to what you can do with the tomato, for example. So that means it's a bigger challenge for us as a system. However, there are some other opportunities. The opportunities are a lot of variation for the trait. So all the peppers come from either South America or Central America, and actually different species, about 5 species of capsicum are edible. And out of those 5, different cultures have adopted certain type of fruit, and then that became important in that particular country. So if you go to different part of the world, people consume different type of peppers, one more than the other. I put in here a picture to show the kind of variation that's there. It's really beautiful, lots of colors, lots of flavor differences, lots of size differences, so many differences that are there that relates to all kinds of quality traits. So this is why the interest is, as a geneticist, all of this variation is really cool because you have lots of genes that you can play the game with and then mix and match and create new types, new varieties. So we call them specialty peppers. A specialty pepper would be either could be sweet or hot pepper that's used for special culinary purpose. So that means you're using it for making a particular dish and it's got a particular flavor. And you have all these many different varieties that I listed just here to show you the type of things like cayenne pepper, jalapeno pepper, hatch pepper. So these are kind of things that are available that are all different flavors and different uses for those peppers. So that means we have a very rich biodiversity to start with. So if you're a breeder, there's nothing better than that because you can play the game better. And I thought this would be a useful thing for teaching students how to create new varieties. And then the problem is most undergraduate students come into the program for only 1 semester because they don't have enormous amount of time to do research like a PhD student who will be spending 3-4 years, like 4 years, 5 years on a particular project. So then what happened is that I would like to then create a project that's going to go from 1 semester to another, from one group of students to another. So the idea is to generate a project, generate the idea first, and then tell the students to come in and then continue. It's like a research relay. So that means like 3 students may be working this semester on a particular topic. They will create all the seats, wherever they are, they're going to stop that particular semester and then hand over the seats to the next group of students who will continue. So this way the project is never ending. And so I started this project in 2010 and it's been now 10 years and we have done lots of different things and that's really exciting. However, instead of telling all of the different projects we have, I just only want to concentrate on 2 small pieces of work so that you kind of get an idea of what we are doing. So in this case, our strategy is that first we need to generate all kinds of inbred lines that have interesting character combinations. So we will just create stable lines that are good. So one of the goal is to creating inbred lines of a variety of different characteristics. And then we will release some of the inbred lines and some of the F1, we will generate some hybrids and then we will release it as new varieties. That's the second goal. So this is one part of the goal, which is the product pipeline. The other pipeline is to do some functional genomics work to understand how different traits in this particular species are. For that, we are using 4 different types of work. And one of them, we use a technique called RNA-seq. This is to identify transcript levels of different genes. So this kind of looks at gene expression. And the second thing is DNA fingerprinting to create DNA-based markers, because these markers are very easy to use. In the long run, it would be very useful to link a marker, DNA marker, to a characteristic so that we can do better selection. And then what's called a quantitative trait loci, or the regions in the genome that controls a character, but these are multi-genic loci, so that could be several genes controlling one particular trait. And so we identify these QTLs by using mapping populations and then creating transgenic plants where we introduce a foreign gene to test the particular gene's function. These four things, I'm generally calling them together as functional genomics. I'm not going to present any of these four in the pepper work right now. We are doing in many other species too in the lab, but pepper, I'm not working on these aspects. The progress is going on right now, but not yet been published. But I'm going to tell you the stories about some of the inbred lines we created and some of the characteristics that we studied as a sample to tell you the kind of things we do. And both of those are in two different papers. So one of the first paper we did is to evaluate inheritance. This is like a Mendelian genetics inheritance of fruit quality traits like shape, pericarp thickness, Brix. And this is a measurement of sugar content or a proxy for sugar content. So we look at that, and then also a bunch of traits that relate to shape of the fruit, length of the fruit, size of the fruit, those kind of things. Why did we choose all this? Because they are easy to measure, but also they all relate to the final quality of the product. So in this case, the fruit. So that means we have to first understand these characteristics and then also need to generate a lot of data so that we can kind of learn something about those characteristics so we can make our selections much more critically useful. And then make novel combinations of traits, and then hopefully we'll get new varieties. So that's the kind of objective we had. So I'm just going to show you one particular cross we made in 2010. And this is the plants were grown in 2-gallon containers. We put it in a greenhouse. The greenhouse has got a lock and a number lock, and all the students can go on in and out 24/7. That means any time of the day they can go in and do the crosses. And then they have to harvest the fruit and then collect data on various things. So different sets of groups of students were allocated to do measurements on fruit length, fruit width, the weight of the fruit, color of the fruit, and all of that. So I'm going to tell you a little bit about carotenoids. This is what makes the pepper fruit bright red, orange colors. The carotenoids are pigments that are color pigments in the leaf as well as the fruit. But what makes the color peppers brightly colored is from the accumulation of carotenoid in the fruit. So we did some work on that, and that's what I'm going to share you a little bit right now. So this is our greenhouse. You can see the students are potting the plants, and we're putting one plant per container and then growing them here. And I also had a visiting scientist at the time from Brazil who also was involved in this work, and her name is here. And this was done in 2012. So, and this particular cross I'm talking about is with a sweet pepper that's called a round of Hungary. And this particular pepper has got a thick flesh and very somewhat sweet pepper and doesn't have any pungency at all. On the other hand, what's called a Bulgarian carrot is shaped like a carrot, extremely hot. In fact, it's just too hot to use it very easily, but some people like extremely hot peppers, but this is one of them. And then when you make a cross, you get an F1 that has got a different shape than these two fruit. So this is one of the very first crosses we studied. And then we took this F1 and then studied the characteristics to see what the characters are. If you see the Bulgarian carrot is very thin pericarp, 1.68 millimeter, and the round of Hungary has got 4.43 millimeter as a thick flesh. The F1 was somewhat intermediate, but more like round of Hungary in terms of fleshiness. If you see the length of the fruit, this one is 6 centimeter. This is 3 centimeter length of the fruit and 5 centimeter in the F1. And the width of the fruit, 1.34 versus 5.76. The round of Hungary is much wider, and this is the F1. And if you look at the pungency, it's 25,000 Scoville units of pungency here, very high pungency. This one is sweet, and the F1 was similar like Bulgarian carrot. This is a common observation. Anytime we make an F1 with a hot pepper, you get a hot pepper in the F1 because the pungency is dominant. Then the Brix values are low, except the round of Hungary is about 5.2. We look at the Brix at the ripe stage when the color change. And then the color is Bulgarian carrot is orange color, and the round of Hungary is red color, and the F1 is red. So a lot of simple observations. And the next thing we did is to grow hundreds of F2 plants. So every one of the fruit in this photograph, this got F2, each one comes from one member of that F2 population. So every single plant was sampled, and then we collected the fruit. We measured all these different variables at whatever I showed you, similar kind of thing. And then we did that. What's one thing you will notice is there are two kinds of colors. So you have orange or kind of yellowish color or red color. There's two kind of color. But then if you look closely, the color is not exactly orange. There is some that are very orange, some that are less of an orange, between yellow and orange. And same thing here with the red, the intensity of the red also varied somewhat. So there were like two types of color. That led me to think that maybe we should look into the color a little bit more, because in 1950s, somebody did the work on color, how the color inheritance works, red and yellow in pepper. And the red is always dominant over yellow. So those have been resolved. However, I thought there was something more we could do. And that's when we started looking at the color a little bit more in depth. The other thing is that this is a pericarp microscopy. If you just make sections of the wet sections of the fruit, put it under the microscope, you can kind of see how the cells are, and you can measure the diameter of the cell and count the number of cells in the pericarp. We found the Bulgarian carrot, the thin-walled one, had 23 cell layers. The round of Hungary has got 28 cell layers, but also the cells, each one of them, were very large in the case of round of Hungary compared to the Bulgarian carrot. This kind of led us to think that the thickness of the pericarp is controlled by both the size of the cell as well as the number of cell layers. So both of them are controlling it. So probably there are 2 or 3 genes in controlling that trait. We were also interested in finding out what happens this kind of shape that I call pumpkin-like shape, which is that we could call it a ribbed shape. So the fruit pericarp is fully ribbed or slightly ribbed or smooth. If you categorize the population, it came out to be like a 2-gene model. In other words, in Mendelian genetics, the ratios were fitting to a 2-gene model. So both of them, one or both of them showing semi-dominance. So we kind of came up with this idea of semi-dominance. That means the ribbed one is slightly ribbed in this case, that is semi-dominance. So we could kind of categorize them into 3 groups, and it fitted to a 2-gene model. So that's interesting because nobody resolved that particular part of the genetics. And we also did this for fruit shape, and that fitted like a 1-gene model. This is round versus long and intermediate. And this particular type of work has been done before. It kind of confirmed what previous research studies showed. Another thing we did is we look at the data and see whether there's any kind of correlation between 2 traits. So we look at fruit width and fruit weight. And this is like each one of these data points is for one particular plant in the F2 family. All the F2s are in the blue color. So you can kind of see there is some relationship, but it's not a very high quality relationship. R-square value is pretty low. So it's the relationship between width and weight is there, but it's not very strong. You do that for fruit length and fruit weight, there is no relationship at all. They are not related. In other words, fruit length behaved somewhat unusually in the F2 population. One thing we notice is that we get a lot of transgressive segregation. So that means you would find actually longer fruit. There are some that are 100 millimeter length that were longer than the parents and the F1 and all of the F2, mean values. So they're much more than here. So you can see the midpoint is around 50 somewhere here, but we are getting some of them are about 100 millimeter long. So that said that somehow the fruit length is controlled by genes that have this transgressive segregation. So what must have happened is that the genes for length of the fruit must have come from more than one source, and then they are interacting in the F2 population. So certain plants have much longer fruit. Now, what does it tell you as a breeder is that if I take plants that have this very long fruited lines, F2 lines, and then self-it, I will be able to get lines that have this long fruit, which is very easily breedable, like transferable in that. So we also looked at total soluble solid and mass of the fruit. The relationship was poor, not a very good relationship. Weak relationship was there, particularly when the fruit weight was bigger, you get a slightly higher Brix. But in peppers, we get about 5 Brix in a ripe fruit. That's a good quality. I mean, it's a nice amount of sugar in it. Next, this color value, I told you about colors. And instead of just reading the color by eye, which we can do as yellow and red and all that, you can use a machine to read the color. So we use a Kodak color meter, and the Kodak color meter reads the values, and these values are designated as a B. For example, positive B values are referring to yellow color, negative B values are referring to blue color. And same thing here, red versus green. Positive A values are red and negative A values are green. So we use this machine to just collect these values. And the beauty of this is that these values can tell you the intensity of that color, within the red color, how intense red it is and all that. So when we did that, this was interesting. If you look at on the x-axis, we have red color space, that's its positive A values. And then here is the yellow color space. This is also positive B values. When you correlate these two, there was a set of fruit that were all red. They were all clustering together. So every one of these red ones, as you increase the red color, you're also increasing the yellow color to some extent. The yellow values are increased there. However, there were lots of different yellow color fruited ones that were not correlating like this. They are dispersed all over the place. These are all the yellow color fruited ones. But what's interesting is the Bulgarian carrot, that one of the parent was yellow or orangish yellow, and that also had values that are high values like 60 yellow color space values. However, when you look at the x-axis, that is 30 A unit, I kind of wondered why should this give an A value? Because the yellow peppers are yellow, we think, because it doesn't have the red pigment. So why on earth this is measuring 30 A value? That really meant the machine is seeing some red color in the yellow pepper, and I can't see it by eye, but there must be something red in there. And that was one of the questions I raised. I said, well, there must be a red pigment in there. To tell you a little bit more, I got to show you the carotenoids that are made in the fruit. That'll be the next one. But before going to the next slide, I just want to show you here is that the yellow color was also related to color lightness, which is also measured by the machine as a YAL value. So as we increase the yellow color, we also increase the color lightness. Color lightness makes people perceive the fruit as bright instead of dull fruit. You kind of look at it and say, wow, this is bright yellow or bright orange. That brightness is by increasing yellow color. So now the color pathway, where do these color pigments come from? The color pigments come from they are synthesized, they are carotenoids. The carotenoids are long chain compounds, and there's got lots of, the pathway is in the plastid. So the plastids that are making the colorful carotenoids in the fruit are synthesizing it upon when this ripening is starting. So when the ripening is starting, the pathway is really working very high levels of flux, and it starts with the phytoene, and then you have different kind of carotenoids made. Up to here, this is the lycopene. The lycopene is the red pigment, the bright red of the tomato. So phytoin-2 lycopene, and then this lycopene is converted to alpha carotene or beta carotene. And the beta carotene is a very important carotenoid in our diet because we convert the beta carotene to vitamin A in our body after we consume beta carotene rich food. So from here, beta carotene, we have cryptoxanthin, zeaxanthin, anthraxanthin, violaxanthin. These are called the xanthophylls. These are oxidized version of the beta carotene, and these xanthophylls are important to protect the tissue from high light damage. So there is what's called a xanthophyll cycle. There's a conversion back and forth between zeaxanthin and violaxanthin. And some of this violaxanthin is also a precursor for an important growth regulator called abscisic acid. So from here, violaxanthin to neoxanthin, neoxanthin to ABA. This pathway has got multiple steps, and I have just reduced it in this pathway. So that means you have a very important compound, beta carotene, important in our diet, important in the physiology. But in peppers, there is something interesting happens. Some of this anthraxanthin, pretty good amount of anthraxanthin, is converted to a compound that's called capsanthin. Capsanthin is a red color pigment. And also another compound from violaxanthin to capsorubin. Capsanthin and capsorubin, both of them are bright red. So all of the red peppers accumulate capsanthin and capsorubin. They don't accumulate lycopene. Very smaller amount of other carotenoids, but capsanthin, capsorubin is a huge amount, and that makes a red pepper red. And is it any use for us? This is not nutritionally not valuable because we're not concerned. We're not converting capsanthin to vitamin A or anything. Beta carotene is more valuable in that sense because it's a nutritive carotenoid. This is a non-nutritive carotenoid, but some of this is also esterified to fatty acids, and a whole bunch of compounds are created that we call xanthophyll esters. Currently, my research in my lab, we are looking at this particular pathway and some of the genes that were not previously characterized. So we're looking at the function of these different genes in this pathway. I'm not elaborating that particular work, but I'm just going to show that capsanthin, capsorubin is what makes the red pepper red. And now I had this idea that maybe that Bulgarian carrot, the yellow orangish pepper, should have some red color, is what I suspected. So we extracted it and we did a HPLC. This is a HPLC run. And then this was connected to mass spectrometry to identify the compounds. So what you're seeing here, these two peaks, capsanthin and capsorubin. The first peak is capsanthin. The second one is capsorubin. So these two peaks are the red pigment. And then here, round of Hungary, the red parent, if you see capsanthin, that's capsorubin, there are two peaks. So what it is telling us is that I suspected initially, based on the color values, that the yellow pepper should accumulate red pigment. And we proved it because when we did the analysis, we could actually show pretty good amount in terms of total carotenoid, pretty good amount of red pigment in that yellow pepper. So what it really told us is that something is perhaps reducing the color of that pepper, the red color of that capsanthin and capsorubin in that Bulgarian carrot. So we're not seeing it, but the machine is seeing it, both the color reading machine as well as the mass spectrometry proving that it is there. So this was a discovery in a way, because people previous to our work thought that you can get the yellow pepper when you have a mutation for making the red pigment. Because if you stop this pathway here, for example, if a gene is mutated here, this is a capsanthin synthase, this particular gene CCS, if this gene is mutated, we will not get the red pigment, but we will accumulate beta carotene and other yellow pigment so that the fruit will be yellow. That's one way we can get that kind of yellow peppers. But now we proved that the yellow condition could be also when in the presence of this, but we can still get yellow. Now, the nature of that genetic nature of what happened there, we are still inquiring that. But whatever we have so far done in this first piece of work has been published. And some of the things we learned here is about the pumpkin-shaped corrugated pericarp fruit shape, something about pericarp thickness. We have discovered some new information, and about something about Brix. And we also did a lot of inheritance data on shape, color, morphometric trait, length, and width of the fruit. But those things all confirmation of what previously known knowledge. And then we also did this color space measurement, and that led to some carotenoid analysis that said that orange yellow fruit already have some red carotenoid, although that is not red for our eyes. So we are using the progeny of this particular cross to develop some mapping population to map genes, and that's going to be some of our future work. Now I'm going to move to a second part of the work, and I'll do that very quickly, but if you have any question at this point, I can answer that. It looks like someone raised a hand, and I will just stop. Okay.
After completing this second, we can have a question session, sir.
Very good. Thank you. All right. So I'm going to tell you a story about what we did with a particular line. I'm calling it a UF RJ1076A. All of this is an inbred line, how we developed it. Again, the Round of Hungary, the red fruited variety, that's what we used for a cross. We crossed it with a jalapeno pepper, which is a hot pepper, a pungent one. And so this particular pepper then gave rise to an F2 population. I had about 200 of these plants, and all these F2 plants were numbered 1 to 200, and they were inside a greenhouse. And that particular season, we had a major problem with the mites. Broad mite is a problem, and the broad mite attacked lots of plants. So I kind of lost everything, almost everything. Some 5-6 plants were surviving that particular outbreak, and because of that, and because they looked somewhat better than others, I kind of saved. And one of them, which was the plant number 107, was the one that I liked most. I said, okay, I'm going to save seeds from this and continue with this. So this is really a chance event that happened. Then I selfed these plants, that particular RJ107, and then whatever progeny we got were studied for fruit quality trait, just like you heard in the previous section. So we were collecting data on fruit quality. So we had F3 and then F4, and we continued. And then each time we were, after doing the first work in the greenhouse, we took it outside the greenhouse and we started growing them in the field. So each time we will only select one plant and then number that, and then that will be advanced. So this is what's called a single seed descent method of breeding, where you're selecting only one and then advancing it further and further. Okay. So at that time came, so this is the whole sequence of events. Round of Hungary jalapeno that were crossed to create the F1. We created F2 population. We forwarded it to F3, 4, 5, 6, and 7 generations. After the 7th generation, we got these inbred lines. A whole bunch of stable inbred lines were obtained, and those are all good for us in terms of characteristics. But one of the things we did was that these plants were all grown in a field, and here the soil is sandy soil, and we use what's called a drip irrigation and a plastic mulch. So that's where the system where we grow. You can kind of see the peppers are pretty nice, big fruit. And these fruits were harvested, and we measured the yield and other traits. One thing interesting is this field contains a nematode, which is called a root knot nematode. Now, I didn't start the work saying that we're going to breed nematode-resistant varieties. However, at that time, I was thinking about introducing nematode resistance into our breeding lines. So our initial plan was pretty simple. Initial plan was pretty simple, is to take a nematode resistant line and cross it with our lines. This was the idea. But before that, I'm just going to show you some data regarding fruit quality traits. And so in the next slide, what you're seeing is a student working who is just collecting data on the fruit quality. So this will be like a pericarp thickness or weight of the fruit, length of the fruit, those kind of things. And then we have collected data on a variety of different ways. One way we collected data is using a scanner. So there is a program that is free program called Tomato Analyzer. In Tomato Analyzer, you can analyze the shape of the fruit by scanning the fruit, like you slice a fruit and scan it, and then that will provide data on area of that fruit cross section, and also the length and the width and so many other things, some shape parameters, and then it gives you all these different parameters. You don't need to read all these numbers, but I'm just going to tell you the first column, JAL, is one of the parent and ROH is the other parent. Every place there is this highlight is that some of the lines that had characteristics that were significantly different from either parent. So we are creating a new type of shape or new type of shape related parameter that has been generated. So you can see however many, because all of these lines are all coming from that single one plant that I chose in the F2, which is RJ107. But you can see RJ107 11K, RJ107 8I. So each one of these is different progenies that are derived from it, but within that, it's segregating for the shape. So what's one of the very interesting thing with the pepper is that the fruit shape is controlled by multiple genes, and there are lots of genes that have not yet been characterized, and you can get really fine differences in shape, very small aspects, but they are definable, they are inheritable, and all of that. So that's what we have shown in our work. We also did this color space value measurement, similar like that, and some of those values that are shown here are showing they are different compared to the two parents, and those three lines are differing, significantly different from either parent. And then you have here, we did a test, and to test hot peppers is kind of hard because people have to chew it, and then it's going to be too hot to taste, and it's not a good way to taste it. So we kind of came up with a recipe that is mixed with some lemon juice and tomato. We call it a salsa, and then that salsa was eaten with chips, and then you score it. So here is a panel of 85 people who evaluated them for different things about overall liking, flavor, texture, spiciness, and this type of and ranking. And this kind of scale is called a hedonic scale. So using this hedonic scale, we came up with this ranking, and we found out this particular line has got distinct marking. So one was the 6B and the 6A. Both of them were liked more than the other lines by this particular panel. Then came this nematode resistance, and I didn't start with even, our goal was that there are lots of nematodes in Florida, and these nematodes, root knot nematodes, are kind of meledogine species, and this one of them is called Incognita, Arenaria, Javanica, Hapla. All of them have different species, but they have many races within them. Within these races, it's not very easy to figure it out unless you test them on a bunch of different plants. So you can see here it's a tobacco, tomato, pepper, watermelon. So you put them, and certain races will be sensitive for some varieties will be sensitive for one race, but not the other. So you kind of distinguish them based on whether something is resistant or something is sensitive based on that. So we have these different races, and so we chose to work with the Incognita. And the initial idea was that previously existing USDA variety is called Charleston Bell, which is a root knot nematode resistant bell pepper. We'll take this bell pepper and cross it with the pepper that we developed in our program. After that, we will examine and screen for resistance and select for fruit quality, and then also with the root knot nematode resistance. So that was the plan. So we started out doing that or thinking about that. Then we thought, oh, first we have to check our inbred lines, how susceptible they are, how resistant they are. So we did a test, and those tests were done in these kind of sand cultures. You grow the plant in a sand in a small container, and then you infect it deliberately by using juveniles that are M. incognito juveniles. And then you let it grow for a month. After 1 month, you can then take the root out, wash it, stain it. And when you stain the roots, you can kind of see where the nematode has reproduced. This is called the egg masses. You can actually count these egg masses, and that will be a quantitative way to know how much infection happened on the plant. So we did that and this type of analysis. And also you can stain it in certain way and see actual nematodes inside the root. If you look at that, wherever you see this dark staining inside, there are tiny nematodes. There are many different nematodes have gone into the root. So you can look at that penetration of the nematode into the root. You can examine that. So with such studies, what we did is we compared a bunch of different varieties. And so we found out, to our surprise, one of the lines that we already developed had a very high level of resistance to root knot nematodes. So you can see here, this is the number of egg masses per gram fresh root. So this one is like UFRJ 1076A3. It's got like a 0.2. That's a mean number of egg masses per gram fresh root. So if I have a dozen plants, one plant will have one egg mass in it, and that's it. And they don't want to, the nematodes don't infect that. However, if you see these other varieties, and the Charleston Bell is the standard that's supposed to be resistant, in our assays, they were not resistant. So we were not sure what's going on. In fact, repeatedly we have shown for that particular race 3 in Melanogain incognita, that particular variety turned out to be sensitive. But then there are also California Wonder, which is known to be a very sensitive variety. That's had got twice as many egg masses than the Charleston Bell. So there is a gradation of resistance, and some of them have more resistance, some of them are less resistant, some in between. So we calculated other parameters like a number of eggs per gram fresh weight. There is something called the egg mass index, the number of egg masses per root. There's what's called a reproduction factor that takes into account how many juvenile nematodes that we inoculate and then how much it has reproduced. So by counting that, we look at those numbers. By all different criteria, we could say that the inbred line we have already is very resistant to root knot nematode. So this led to our work on additional nematodes. So these are different species of nematodes for which we check the resistance. MA is M. arenaria, incognita, Enterolobii, Floridensis, and Javanica. So these are different species of Melanogain that exist in our production system. So we kind of scored for that. What's interesting is that none of our many varieties of we screen, none of them is resistant to Enterolobii. And that is very well known in the pepper literature, that nobody has found a resistance to a pepper plant resistant to Enterolobii. But for other species, there is some resistance known. But in this case, you can see that the UFRJ 107 always was very resistant to arenaria and always very resistant to incognita. And on the other hand, California Wonder, which is a variety that is sensitive variety, had got a very high infection by this arenaria and incognita. And however, if you see the UFRJ at 0 or 570 versus 11,000, this is the number of eggs per gram fresh weight. So this actually tells us that, well, we have a very good line that is resistant. The Floridensis did not infect in both of the peppers. The Enterolobii was susceptible, like both varieties are susceptible to Enterolobii. And Javanica, again, there is some amount of, there is no difference here, but some amount of resistance, or relatively more resistant. This line was more resistant than California Wonder. So this is what we have data-wise, that the root knot nematode resistance. We were interested to know, where did we get this root knot nematode resistance? And so we went back and looked at the parents, and the parents were not resistant. Somehow in the RJ107's progeny, that one plant I picked, some of the progeny lines were actually gained the resistance. So it looked like, based on inheritance studies, it behaves as a recessive trait. So that's also interesting that we got a recessive trait. So in the literature, there are other genes for root knot nematode resistance, and those lines are known. And so we don't know whether the gene we identified, the resistant we identified, is the same or allelic to those lines, or is it something new? That part we don't know. So those are some of the conclusions that jalapeno and pre-release germplasm, this one displayed high level of resistance to incognita. The jalapeno is one of the parents, so that parent was slightly better than the other parent, was very, very sensitive. This one is somewhat resistant. However, the line that we developed actually has much, much more resistance. And then nematodes penetrated the roots of, so we also did some penetration studies by looking at the staining one, and we couldn't find anything inside the root of U of RJ 1076A, but then we found it in others varieties. None of the lines were resistant to enterolobii. That's because we don't have that, and that particular species of root knot nematode behaves slightly differently. So the final conclusion is that the germplasm we developed, the pre-release germplasm, the inbuilt line, is suitable for root knot nematode resistant rootstock because we can graft the sweet peppers onto this. If you graft a sweet pepper on a rootstock of a hot pepper, the fruit will be like the scion. In other words, it'll be sweet pepper, no problem. However, the root will contribute to root knot nematode resistance, which is a good thing for us. So work I described, both of them have been published. And you can see this has got so many authors because so many students worked in this work. And sometimes it's even not horticulture students who work on these inheritance studies. It could be Japanese major or English major or people studying, law, and they come to the lab and try to learn something and do. So this kind of interesting experience training those students. And then they did that. This work, actually many years of work together, was published in 2015. And the work on the nematodes was done by a student. He was an undergraduate student, but he worked with a postdoc, McKillen, and then developed that work, and that was published this year. We are doing some more work on the metabolic targets, the carotenoid biochemistry. So we're looking at that in our lab, but those are not completed yet, the genomics work. But when completed, they will be published. So I call this project Building Better Peppers, which started out with a mini grant for teaching. So this is part of our teaching, hands-on teaching to teach horticulture to students. So this is from the College of Agriculture Life Sciences, so that funded a mini grant to start the project. After that, I have been supported by a USDA agency, that is Florida Department of Agriculture and Consumer Services grant, and that supported our work on the nematodes, the most recent work. So with that, I would be happy to take any questions, and I'm going to leave the slide here. I would stop at this point for questions.
Participants, you can raise your questions in a chat box, or if you raise your hands, we can connect you to with the speaker. So I think Suresh Veera, sir, please wait for a few minutes. We will connect you. Connect one. Mr. Suresh Veera, you can connect your audio.
You may have to unmute, I guess.
Yes.
Is there any relationship between pollen color and
Sir, we have questions received from Mr. Gnanasegaran, and he has asked, is there any relationship between pollen color and pepper's fruit color?
Within Capsicum annuum, there is not such a relationship, but there are different other capsicums that have different colors, pollen color. So I haven't examined that really. However, we have a project where we are trying to create multiple colored peppers. And in that project, we are finding some interesting things, but I can't tell you exactly now the pollen color and this to your question. However, that's one of the characteristics we are collecting data on, because some of them are greenish, some of them are purplish, and that's a useful trait.
Okay, sir. And one yes, Preeti has asked, can Scoville units be enhanced by crossing?
Yeah, that's possible, yes. The interesting thing with the Scoville units are it's kind of difficult to measure. The amount of capsaicin for doing that, you have to extract it. And what we found out, the problem we found out is we are quantifying the capsaicin using mass spectrometry. And for that, you have to first extract the fruit and then take the extract to a unit that does the mass spectrometry. So the very hot fruit is not easy to handle because when you're grinding it, the whole place becomes very hot. Everybody is sneezing. So it's kind of a difficult work to do. So we haven't followed up on the capsaicin content more and more. We know by experience, yes, some fruits are much more intensely hot than even the parent, but we haven't done the actual work of measuring it. We have done very small amount of work on capsaicin for that matter.
Thank you, sir. And Ms. Saundarya has asked a general question, and she would like to have insights on ethical aspects of breeding or crop improvement in research.
Okay. That's a good question. So now all of the vegetables and fruits that we are consuming have come from some kind of breeding that are simple selection that was done hundreds of years ago, like inbred lines and seeds that have been saved by farmers and all of that. So the ethical question comes in when we are doing transgenic work, when we transfer a foreign gene into the plant. Now, the question is that some people think, or their argument is that, is it fair to take a gene and put it into a new plant that never existed before? Is that fair? Because are we playing God by doing this kind of creating a new transgenic plant? That is the argument. My argument is that the transgenic plant itself is not bad because we are introducing a very specific known gene into a new genome. So if I am transferring a tomato gene into pepper, I know enough about the tomato gene. I know what it codes for, what kind of protein it makes, what kind of product it is going to influence. So that's there. However, the people who are opposed to GM product or genetically modified plants, they are opposed because the technology itself, they are opposing the technology. Instead, I'm thinking that we have to go by product. So for example, I can take a gene that is negative trait gene, like something, say, that is not good for us, and put it into a plant and make a plant that is not useful and that maybe even toxic. One can do that. So at the same time, you can also improve a plant for a good, for a healthful value. For example, if I know a plant has been manipulated to increase beta carotene, I will love it. How that manipulation was done, whether by putting a gene from algae or gene from a tomato, it doesn't matter because I know the end product is the whatever we have improved is improved for beta carotene, which is good for me. So based on the science, I'm going to choose that product than the other product. The problem with the industry is that the industry does not want to label genetically modified product so that you don't know really what what modification was done on a particular variety. So that's a problem. So I feel that it's okay to label it, even have some kind of code so that people can go in and figure out what was modification that went in, so that you can kind of get an idea, well, is this improvement is what I like, so that I can take this transgenic plant, but not the other transgenic plant. That means you have in the market a way to select. So that's a possibility.
Thank you, sir. And there is a question about the protocols in breeding by Agri Consciences. How could RNA sequencing help in the development of resistance for pathogen?
Very good question. But the RNA-seq is a method to study transcript for many genes. We're looking at hundreds and hundreds of genes at the same time. So to apply it in a context, we can look at genes that are, for example, if you have two inbred lines, there are sister lines, but one is resistant to a disease, another one is not. If you compare the transcript of these two lines side by side, you will be able to identify genes that may be highly expressed in the resistant line, but not expressed in the sensitive line. Then the candidate gene may be one of those, not necessarily it is the actual gene. It could be that. It could also be other factors. So, but you have to at least have an idea and say these 6 genes are highly expressed in the resistant line, but not in the sensitive line. And if you know that information, at least one of the 6 genes or many of the all of the 6 genes could be important for that trait. So this way you're going to find that connection. So that's how the RNA-seq can be used. RNA-seq is not an end itself because you can't use it directly in breeding, but you can use it, that information, to make a judgment about what genes are important. So this kind of gives us that idea. But you can also create, RNA-seq creates a lot of data on transcript sequences for many, many different genes. And you can also use that data to identify variation. So you can create DNA fingerprints to identify a character and then a DNA fingerprint to go with it. So the nice thing, for example, if I have a DNA fingerprint that I can identify a plant that is resistant to the nematode, I don't have to grow the plant for a long period of time and then put it in the field and test it, all of that. I can just germinate the seedling, and within a month, I can screen for the DNA markers. And then based on the markers, I can say these 6 plants or these 5 plants are likely resistant, and then do the test on only those plants. So this way it becomes more efficient. The method becomes more efficient.
There is a query from Ms. Kalayarasi. You can directly ask your question, and your voice is unmuted. Kalayarasi, you can ask your question. I think she
Okay.
could
No
not
problem, but
connect, it seems.
in the
Okay.
chat, I see
Sir,
a
please, sir.
question. It says, can altering capsaicin coding gene affect any other character? I don't think so. I have not done such an experiment, capsaicin, modifying capsaicin coding genes, but the genes have been identified, that those are known. So whether it'll affect other things, it's kind of hard to know at the moment. I think people are, I do see a small difference in flavor. The mutations that affect capsaicin content also changes the flavor of the fruit, not only the heat of the fruit, but flavor of the fruit.
And if there is no questions further,
Okay.
we are coming to the end of this today's session. And on behalf of the Annamalai University, the alma mater of today's speaker, and we would like to profusely thank you, sir, for your a spicy, colorful, and thought-provoking lecture on crop improvement in peppers. Really, it has given a nuances of color space value and color pigment and space related parameters of pepper, bell pepper, that is very new to the young budding scientists. And it gave us industrial perspective also. That is the specialty of your lecture, sir. We whatever we teach here, that goes in line with the industrial requirement and in line with the industrial perspective. But it has really benefited all young budding scientists. And more than that, we have got connected with more than 300 scientists across the globe, wherein we have all scientists from across the state agriculture universities of India, and some of them from abroad. And we have industrial partners who have participated in this seminar, and it has given a very nice perspective of breeding pepper with their perspective, and we hope that it is much useful for them also, sir. And more than that, what we could observe from this your lecture is you have given a breeding to stress, biotic stress related aspects, and that works are have very significantly addressed breeding to the agronomic traits. That is what coming up in the industry, sir. That we could able to observe from your work, sir. And it is really a insight to all researchers in India to use that for their future breeding programs, sir. We, on behalf of the Department of Plant Breeding Genetics and Department of Horticulture, Annamalai University, we would like to profusely thank you for your for sparing us your valuable time and your expertise and knowledge, sir. Thank you very much, sir.
Thank you very much. It was
And
an honor for me.
thank you, sir. And I would like to, on behalf of the alma mater, we would like to invite you whenever you find time and you can. We just would like to invite you after all this pandemic period. Sir.
Sure.
So
Thank
we
you
are
very much. I
expecting
will do
you,
that.
expecting to meet you in person whenever you come to India, sir. And
Certainly.
along with this welcome note, our speaker, I would like to conclude the official part of this seminar from the webinar disc. And we would like to thank all our authorities, our Dean, Dr. K. Manivannan, and our Professor and Head of Department of Plant Breeding and Genetics and Professor and Head of Department of Horticulture, and the Director, Dr. Prakash sir, and all other co-workers of this team, and all staff members from both the departments, and all participants for their efforts and for sparing their time to conduct this useful session with us. And we would like to specially thank Dr. Gokula Krishnan sir for his support. And with this, we would like to conclude, sir. Thank you and namaskar.
Thank you very much. I appreciate it.